Referring to the earlier tendency to assume all cancers of a particular tissue have the same root, Brown said, "It was like thinking a stomachache has only one cause." Recognizing the distinctions makes it possible for us to do a better job of treating these cancers. At Cold Spring Harbor Laboratory, Michael Wigler is using the method in yet another way. Rather than adding RNA to a microarray and looking for gene expression, he is adding DNA from cancer cells to create a profile of the genetic diversity present in tumors. Many cancers are caused by chromosomal rearrangements, such as might occur when segments of a chromosome are inadvertently duplicated, leading to an excess in the number of genes that code for growth-promoting proteins. Other cancers arise due to the loss of genes coding for proteins that repress cell growth. Applying Wigler's technique, clinicians biopsy cancerous and healthy tissues from the same person. DNA from the cancerous tissue is chemically tagged with a red dye, while the DNA from the normal tissue is tagged green. DNA microarrays containing all 35,000 of the known human genes are exposed to a mixture of the two samples. Like mRNA in a standard microarray experiment, the labeled DNA molecules bind base pair to base pair to their complementary sequences in the array. Genes amplified in cancer cells are marked by red spots because there are many more red-tagged molecules binding to that spot than green-tagged ones, while genes deleted in cancer cells show up as green spots on the microarray because there is no red-tagged molecule to bind there. Such experiments have already greatly expanded the list of genes known to contribute to breast cancer. Whenever we tackle a specific human disease, we realize the extent to which we are probing in the dark. We could move so much more quickly to the heart of the problem, and know the exact nature of what is wrong and how we might fix it, if only we had a more detailed knowledge of how our genes express themselves when all is well. With a fully formed dynamic understanding of when and where each of our 35,000 plus genes functions during normal development, from fertilized egg to functioning adult, we would have a basis of comparison by which to understand every affliction. What we need is the complete human transcriptome. That is the next holy grail of genetics, the next big quest in need of superfunding. In the short term, a likely or even more important objective will be to obtain the complete transcriptome for the mouse, whose advantage over humans is that we can both observe and intervene experimentally during the course of prenatal development. Even collecting all such relevant data from the mouse will require major investments of money and time. And as proved by the experience of DNA sequencing, we will be well served to take the time to gain what expertise we can by completing transcriptomes for simpler model organisms before taking on the mouse, much less the human. Microarray studies of gene expression during the yeast cell cycle have already revealed the staggering complexity inherent in the molecular dynamics of cell division alone. More than 800 genes are involved, each called into action at its precisely specified time in the cell cycle. Here, too, we may depend on evolution's reluctance to fix what ain't broke. A biological process, once successfully evolved, will likely continue to employ the same basic molecular actors for as long as life persists on Earth. As far as we can tell, those same proteins that direct development through the course of the yeast cell cycle carry out similar roles in the human cell. Ultimately, the goal of all three omics, gen, prote, and transcript, is to create a full picture, detailed right down to the level of the individual molecule, of how living things are assembled and operate. As we have seen in even the simplest cases, the complexity is bewildering, and despite the spectacular progress of the last decade, there remain many daunting challenges. As they relate to complex organisms, the molecular underpinnings of development, that extraordinary egg-to-adult journey that is governed by a linear code strand composed of just 4 letters, are for now best understood in the case of the fruit fly. The fly has, of course, been the focus of intensive genetic investigation ever since its adoption by T.H. Morgan. And through the ensuing years of continual innovation, Drosophila melanogaster has remained a genetic goldmine. In the late 70s, at the European Molecular Biology Laboratory in Heidelberg, Germany, Christian Janni Nusslein-Volhard and Eric Wieschaus undertook a spectacularly ambitious fruit fly project. They used chemicals to induce mutations and then looked for disruptions in the very early embryonic stages of the fly's progeny. Classically, the quarry of the fruit fly geneticist was mutations affecting adults, like the one Morgan found to produce white rather than red eyes. In focusing on embryos, Nusslein-Volhard and Wieschaus were not only condemning themselves to years of eye strain as they stared down microscopes in pursuit of those elusive mutants, they were also venturing into utterly uncharted territory. The payoff, however, was spectacular. Their analysis uncovered several suites of genes that lay out the fundamental body plan of the developing fly larva. The more universal message of their work is that genetic information is hierarchically organized. Nusslein-Volhard and Wieschaus noticed that some of their mutants showed very broad effects, while others evinced more restricted ones. From this, they inferred correctly that the broad effect genes operate early in development, at the top of a switching hierarchy, while the restricted effect genes operated later. What they found was a cascade of transcription factors, genes switching on other genes, that in turn switch on others still, and so on. Indeed, hierarchical gene switching of this kind is the key to the construction of complex bodies. A gene producing the biological equivalent of a brick will, left to its own devices, produce a pile of bricks. With proper coordination, however, it can produce a wall, and ultimately, a building. Normal development depends on cells knowing where they are in a body. A cell in the tip of a fly's wing, after all, should develop along very different lines than one located in the region that will give rise to the fly's brain. The first piece of essential positional information is the simplest. How does the developing fruit fly embryo know which end is which? Where should the head go? Bicoid, a protein produced by a gene in the mother, is distributed in varying concentrations through the embryo. The effect is called a concentration gradient. The protein levels are highest at the head end and fall off as you travel toward the rear. Thus, the bicoid concentration gradient instructs all cells within the embryo as to where they fall on the head-to-tail axis. Fruit fly development is segmental, meaning that the body is organized into compartments, all of which have much in common, but each of which has some features unique to it. In many respects, a head segment is organized just like one in the thorax, the middle part of the insect body. But the former has head-specific organs like eyes, and the latter thorax-specific ones like legs. Nusslein-Volhard and Wieschaus found groups of genes that specify the identities of different segments. For instance, pair rule genes encode transcription factors, genetic switches, expressed in alternating segments. Pair rule gene mutants result in an embryo with developmental problems in every second segment.